Same as last time. I'll be sending right now the link to the short quiz to the chat, so which we'll be using for as sort of a snapshot of the opinions in here. Yeah, and I will repeat this the link I think in a short while for the other people to join. Yeah, so I think we can slowly begin today's meeting of our discussion club. And today we are discussing pipeline development and different languages which can be used for this purpose, and basically different strategies and different reasons to describe pipelines using specialized languages. For those of you who have not visited our previous meeting, I will shortly repeat that the main topic of this seminar is the discussion of best practices and talking about different questions and topics. So it's not going to be like a lecture or a tutorial, but we would like more people to participate in the discussion to basically formulate a set of like best practices and how different people do different tasks. And one more thing which I would also like to tell you before we proceed to the actual topic of today's meeting is that our institute is also hosting or organizing a bioinformatics hackathon, which will take place in Yerevan, Armenia this summer in August. And the registration is open until 15th of June, so basically a week from now. And if you are interested in participating, unfortunately, the participation is expected to be on-site only, and online participation will be possible only for people who submit the projects to the hackathon. But if you're interested, you can proceed using the link which is shown here or the QR code and register for this event as well. And this is also we are pretty excited about that. Okay, so now let's move on to the actual discussion. So today we have a little bit shorter set of people on the expert panel. Unfortunately, we hope to have more next time. So today with us here are the same as last time. First, Roman Chernachik from JetBrains Research. Roma, are you here?
Yes, I'm here.
Great, yeah. And also Gennady Zakharov from EPAM System, also a little bit of sound check. Gena, can you hear us?
Yeah, I can. And do you hear me?
Yeah, perfect.
Yeah, perfect.
Good, yeah. So we are expecting some people may join us a little bit later, closer to the end of the seminar, including Alex, who was on the expert panel the previous time, and maybe some other people. Yeah, but basically, most will be talking today with Roma and Gena. For those who have joined recently the seminar, I'm repeating the link to the forum about the today's topic, and basically. We'll be switching and looking at the results shortly. So first, as always, would like to introduce the topic and what we're going to be talking about for today. So today's discussion is about development of bioinformatics pipelines. And I think that most of the audience are probably familiar or pretty familiar with the idea, with the concept of a pipeline. But I think I can introduce this concept a little bit in more detail. So what do we consider to be a bioinformatics pipeline? So a bioinformatics pipeline is essentially a sequence of computational steps which is performed for a particular kind of data analysis. So there are all sorts of pipelines in bioinformatics, and basically the most natural examples, of course, are things like variant calling pipelines, which process the raw next-generation sequencing data to generate genetic variants, sets of genetic variants. But there are all sorts of other kinds of pipelines. So, for example, we might think of a pipeline dedicated to the analysis of gene expression data with RNA-seq, for example, detecting differential expression and running some sort of downstream analysis. We may consider other kinds of analysis for example, genome assembly, especially many people who are working with routine assembly of multiple genomes. They'll have a sort of their favorite assembly and evaluation pipeline, and all sorts of other tasks in bioinformatics. Nearly all of them may be formulated in the sequential form when you have a different sequence of steps which are performed one by one. And on the right-hand side, I have the picture, which is basically taken from the Genome Analysis Toolkit best practices. So that's a set of steps which is used to perform that exact step of variant calling, which basically includes several different stages, and these stages are also separated in like different portions with processing of raw read data to generate and to align them onto the reference genome and to pre-process them to generate basically analysis ready read data. Then do the variant calling, which in GADK is performed according to best practices, unless in the cohort mode, but different options, of course, are available. And there is also another step, which is more about the evaluation of results. And this is, again, a schematic diagram, which basically just exemplifies of what the pipeline may look like. And the main idea, I think, is, again, familiar to most of you, but I'm not sure to what extent people in the audience are familiar with specialized solutions which are developed in order to construct these bioinformatics pipelines and in order to execute them. And having said that, I think we can switch to our quiz and look into the results of our form to see what basically is what do you say about that? So as usually, we are asking about the experience. So for today, we have only people who have some sort of experience in bioinformatics. That's a pity. I think that we do not have people who have not yet joined the field, but at the same time, this makes our results a little more reproducible. And in terms of whether or not people in the audience use the actual pipelining solutions, other methods which are developed to design, implement, and run the pipelines. I think the majority of the audience who have participated in the quiz actually answered that you've had an experience of working with bioinformatics pipelines. You have implemented this, but it's not a part of what you do routinely. But naturally, yeah, there are people who do all sorts of bioinformatics analysis using these special pipelining solutions. And there are people who have never used such solutions in their practice, which is also pretty natural. And the majority of people, again, use pipelining languages, not always, not to do all of their tasks, but to do some of their tasks. And this is a good starting point for our discussion, because this indicates basically that not all of the people use these things routinely. And having said that, I'd like to ask my first question, which basically is about what is the motivation for people to choose and to switch from basically just a collection of shell scripts, which can be used for data analysis, switch to writing a specialized workflow using specialized languages, which we are going to be covering in the next section of this meeting. So what is the reason basically to take these scripts which are used for analysis and implement a workflow using these formal specialized languages? What do you think? I think we can start with Roma for today. So Roma, can you share your opinion on what is the motivation to do so?
Okay, hi everyone again. So for me, the main motivation is it's cluster execution. So special languages allows you to do it in a more convenient way, or utilize your CPU and other sources more effectively. So especially if you use some cluster with submission system with multiple nodes, so your code will be much clearer compared to your bash scripts. And maybe the other way, the other reason, if pipeline is the multi-step, so you have lots of steps and several samples. When you use bash scripts, at some point it's not very easy to implement some complex logic when you match files or generate some file names, and it's easier to use some more natural programming language like Python in a Snakemake. So I prefer Snakemake. So this is two main points from my point of view.
Okay, yeah, thank you so much for that, for the opinion. So Jana, do you have anything like to add? What is your motivation? I think you were the one who answered that you basically implement everything you do using pipelining solutions. So if that's correct, could you please comment on why you do so?
It's correct. And the only one comment is to which extent. I believe everything which is possible, because some things when you need to make some complicated conditions and something like this, it's not really useful to use the pipeline languages. But yes, I'm trying to use pipeline languages as much as possible. Even if I'm using bash, I'm trying to make some script which can be redone next. And the most motivation, maybe the most important motivation is following, that if you think that you will write some one-time scientific, one-time code, just analyze data and forget about it. So all my experience tells me that this is very, very wrong. That's a very artificial situation when you will run some one-time code and will never return to it. Always there will be some additional data, new options to use, some change of pipeline. So you will have to rerun your data, your analysis more and more time. There's a bit different data, there's a bit different processes, but in anything, you'll have to rewrite several times.
Okay. Yeah, so that's very reasonable, I'd say. Yeah, so maybe Roma, could you comment on? So you've mentioned the reasons of why to use the pipelining languages. Could you please comment on whether or not you were the one who answered that you do everything using these methods or not, and if not, then why not? What is basically when is the time to switch to basically transfer scripts into the pipeline?
So it's a tough question. So I do not everything with pipelines because it's require like huge overheads, and usually this is processing this code you use one time. Because I agree with Gennady that it's good when you could reuse your pipeline, but you normally reuse only some parts of your pipeline, and lots of parts are written once. That's why it's reasonable to make a pipeline when you have some fixed approach. So when you want to play with data, with visualization, it's much easier to do it in Jupyter notebooks without any pipeline. But if you want to do some standard things like align many files, like do some QC with some standard tools, and do some differential analysis, it's quite convenient to use pipelines from initial step.
Okay, yeah. Thank you. So maybe then I can tell my own opinion on the matter. Yeah. So basically, here I switch back to my presentation. So usually, yeah, I think all of these things have been have already people have mentioned that, but still, when people talk about pipelining languages, they usually say that they have some like several benefits. And the things which came to my mind when I was thinking about this matter before today's seminar were actually first and foremost the thing that's a sort of a formal description of what you're doing. So my opinion on the issue of why to switch to pipelining languages first is basically that's easier, usually much easier to understand what is going on here. Of course, if you are at least partly familiar with the syntax of how this or that language is implemented, and what are the main constructs, what are the main elements of the syntax. But basically, for example, in Nextflow, we'll switch to discussing the actual languages which are used in the field. But if you go, for example, to the Nextflow website, you'll see there that they are calling themselves basically a project which is a DSL for running all sorts of workflows, and this DSL abbreviation stands for domain specific language. Which is actually a concept which includes all sorts of formalized description of different things. So, for example, you may have a domain-specific language to describe basic biology facts. I'm talking about this because we are having a little experience with this kind of thing lately. So the idea again here is that you have a simplified and a formalized description of things. In terms of pipelines, this is actual processes or tasks. And almost all of the or all of the pipelining languages allow you to basically declare clearly that here goes a particular process or a particular step, which is clearly identified. The inputs, outputs are very clearly set. In most languages, this is more or less evident. And this makes it readable and understandable better compared to a bash script. Of course, there is also the case of reproducibility. And this is, I think, what Gennady mentioned. And this is, in my opinion, also very nice if you are expecting to reuse what you've done. Unfortunately, I am not the best example personally in this case because I am not always transferring my code into pipelines using Snakemake, for example. I keep a pretty large fraction of what I'm doing in shell scripts, and I greatly suffer from that. So that's basically a good idea because with shell script, it's usually harder to make them easily make the analysis easily reproducible, because basically you'll have to include all these sorts of parameters. Of course, you can do that in Bash as well, but that's not as clear as it is in specialized solutions. And it's very easy to forget about something which you will not be allowed, basically, to forget when you are implementing things using specialized things. So that's are the two benefits which I thought about first when I was thinking about the matter. But of course, yes, there is the case of scalability, which I think Roma has already mentioned in terms of that if you have multiple samples or if you have multiple tasks, specialized solutions will basically execute these tasks to make maximum usage of the computing resources which are available, which is also nice, again, if you are processing large batches of data. This is not as important probably if you are processing single samples or several samples. But if you are processing hundreds and thousands, that's of course becomes pretty important. So in our practice, for example, we've had a case when we were constructing a large database of genetic variants from several thousand exome samples. And yeah, in that kind of task with variant calling pipelines for exome data, you have multiple steps which are not efficiently parallelized in terms of that. Let's say if you have 40 CPUs, there will be several steps which are running like ultimately in a single thread mode. So you have to have a pipeline which that particular steps may make use of all of the threads for one task, and for other tasks which are not scaled as simply, it will just like run multiple tasks for multiple samples. So like just make use of all of the threads which are available. So this was a case for which we've really had this this benefit, which was very important for us. Yeah, but summing up all of what I just mentioned, my opinion on the matter is that yes, you should try to use the specialized pipelining languages for anything you do and you plan to reuse. And as Gena said, probably you'll be reusing more than you think. And as I also mentioned, I am the one who is a little bit suffering from my own laziness to go and use a specialized solution instead of the shell scripting I am pretty used to. Yeah, so basically that's my opinion on the issue. So if there are anyone in the audience who would like to tell anything about basically the reasons, the benefits and motivations for switching to pipelining languages, or if you have any questions on this matter, then please go ahead. If not, then we can move on to discuss the exact languages.
Probably I have made a point regarding the Jupyter notebook. To my understanding, sometimes I can treat the Jupyter notebooks as a sort of the pipeline language, because it also allows kind of reproducibility. But what I say about this, that in Jupyter you can execute code from any point, and this is the strong point, and this is also a weak point. Because when you're trying to change something and make different solutions, you will have some like abundant pieces of the code. And if you're using the Jupyter notebooks, you need to periodically clean it, make some cleanup and remove obsolete part of the code, and to be sure that this notebook can be executed from beginning to the end. Because after some iteration, sometimes you can lose this ability. That's what I say about Jupyter. And the next one, it depends. If the most part of the data you process by the Python code, of course it's better to use Python. But in my practice, the most part of my processing was involved in some command line. And when all your Python is consistent in the shell code execution, of course I believe it's better to use Python.
Okay, thank you. Yeah, so if you could next time be a little bit closer to the microphone, it would be a little bit better, because the sound is a little bit not too loud. Yeah, but thank you for the opinion. And the issue of Jupyter notebooks, we'll be discussing that in several meetings from now. And we'll be talking more about the use cases and basically when you have to work with Jupyter and then not. Because the very fact that we are talking about Jupyter in the pipelining section, which is not basically the main use case of Jupyter, is not like pipelining in the sense of using different tools in a sequential way. Because this is more about the downstream data analysis. But again, the fact that it pops up here indicates that in some cases it may be used as sort of a multi-tool to do this as well. So that's an interesting thing, and again, we'll be discussing Jupyter, I think, in the fifth meeting of this series. Yeah, but thank you again for the comments. So with this, I think we can proceed to discuss the actual software and the actual languages which are commonly used to describe and implement bioinformatic pipelines. So let's again switch back to our form. Let's check whether or not there were any like additional responses or not. It seems that there were none. Let's look at this chart over here. Which basically in this chart I asked what is the solutions which you've ever worked with. And in this case, we have, I think, the dominant majority of the people, or almost all of the people who have responded, say that they've used Bash naturally, very easily, because that's the natural way of running the analysis, and the most I think habitual for most of the people. And yeah, of the specialized solutions, I think we have no one who have used CWL. Most of the people who have used any, like 9 out of all of the respondents, say they've used Snakemake. And some people, though much fewer, have used Nextflow in their practice. And having said that, I think for the part of the audience who are not that familiar with this, I'll take a few minutes to basically describe what we are talking about. Yeah, so basically these free solutions, which are shown on like basically the first 3 options in the quiz, are basically the 3 most commonly used pipelining languages, which are, I think, mentioned in bioinformatics. So there are others. I think there are more than 10 today, but the most commonly used ones are Nextflow, Snakemake, and CWL. Our audience uses Snakemake more, which is actually very, very interesting. I think it may reflect some bias of who is participating in the seminar. Because, for example, I think Alex Predils from the Sanger Institute, who was with us last time, who was unable to join us, unfortunately, today, mentioned in one of our private discussions that basically no people at Sanger Institute and around that very, very renowned institution use Snakemake. So all of what they've been doing is actually with Nextflow. And again, in our case, we have a Snakemake as a dominant thing. And the interesting case is that when I was preparing for this seminar, I've run a short Google Scholar citation search to check what is actually the most commonly used solution. And the Google Scholar also says that Snakemake is kind of the most cited thing. With CWL, it's a little bit hard to find out because it is spelled as CWL, like the abbreviation, and this refers to different things. So not all mentioning of the CWL will refer to common workflow language. If you search exactly for common workflow language with 3 words, it yields I think 1,000 mentions across the publications which are indexed in Google Scholar. The Snakemake, it's I think over 4,000. The Nextflow a little less, so I think 3,000 or so. So Snakemake appears to be a little more mentioned than Nextflow in literature, but it's like really interesting how different people, like different audience, answers differently to these questions. Yeah, but this free are the most probably the most widely considered solutions, and they all of these are a little bit different, and this is probably why there is no unanimity in what people choose. There are other solutions as well. I have to mention the workflow description language by Broad Institute. I think the sole use case of which is running genome analysis toolkit workflows. But as we will see in a short while, many people run GEDK functions without this workflow description language, which is developed by basically the Broad Institute itself, which is, I think, quite indicative of that people don't usually like to switch to some new solutions. I think this WDL thing was introduced very as like a new thing, which will be the new standard. But as I've mentioned last time, when people are developing new standard, we usually end up with basically just another solution. So this was the story of WGL as well. But that's at the same time optimized to run some G80K functionalities. And G80K, I'll have to mention again, is mostly used for processing data with the goal of variant calling. That's the main use case. Again, the G80K functionality with WGL works more or less well, but again, not very commonly used for other tasks. Yeah, so having said that, I think I would like to ask again the people who are here with me and with us regarding the choice of languages. I think both Roma and Gennady use Snakemake, but if both of you could comment on what's the motivation for this choice, why do you prefer this over Nextflow, for example? Which I think is gaining popularity more and more recently. That would be very nice. So I think we can start with Gena this time. Yeah, if you are here.
Yeah, I'm here. So what I had some limited expression with CUL and VDL also. Just why I started to use Snakemake, probably because it's simple for local stuff. Because for CVL and VDM, they require to install some engine, then create the pipeline and something like this, and then use some output. And we leave the Snakemake as the smoother stuff. You can just install Snakemake and start using it on your local computer without any limitations. As for Airflow, I haven't touched the Airflow. So probably it just depends what you do first and stuff. Usually, I've mentioned we don't like to switch to the new solutions. And as for the strong and weak points, I believe the Snakemake both strong and weak point over. It's using files and non-explicit dependencies. I like this in construction, but probably nobody knows.
Okay, yeah, thank you. So what about Roma? What's your point? What's your take on why preferring Snakemake for the task to do?
So actually, we did a long path to Snakemake. So first we started like with Bash, but it was okay for formal description, but it was a nightmare if something fails. You need to manually encode all possible checks, and you need to restore your pipeline and clean up data if something fails in the middle. So then we decided to make our own solution using Python, and it works pretty okay for us. At least it was capable to utilize CPU better than Bash. But then we realized that we need to use remote cluster with PBS Torque submission system, and it was not very wise to improve our own solution. So we found out that there is a Snakemake. So maybe Nextflow was also good, but we started with Snakemake, and first we did so we started using it, and simultaneously we decided to make to get a good support of this language in PyCharm. So we also implemented a special plugin for PyCharm that makes developing Snakemake pipelines much easier. And now also we use Snakemake. We don't want to switch to Nextflow. As far as I understand, it's pretty similar compared to different features. And first, Snakeflow was more advanced, but as far as I understand, now they're quite comparable. And for us, we don't want to make new tool for new language support, and we suppose that so there is no good language support, or not smart support for Nextflow. So we decided to stay with Snakemake. So it's maybe my main addition to Ginali's talk.
Okay, yeah. I think that maybe I may use the possibility to add a little bit about this. Yeah, so I am also more familiar with Snakemake. It was not basically the fact that we've chosen this over that for reasons of for some solid reasons, because at the time when I myself started working with pipelining languages, I think Nextflow was at some earlier stages of development, so at least it was not familiar to me at the time. So at that time, I was familiar with Snakemake, and I was familiar with CWL. And the thing about CWL compared to Snakemake is that it basically uses a different paradigm. So I think, again, for those in the audience who are less familiar with how these pipeline description languages work, I think you can say a few words about that. So the main idea, again, of all the languages is that you have several steps or processes. In Snakemake, that's called rules. And they are joined into a sequence of steps, and one step is executed after the other. And the languages basically differ in what do they use to determine this order of different tasks, of basically what process should be executed after what. There are different paradigms. So, for example, there is so-called explicit description, when you set specifically that this process uses the output of the previous process to do basically for execution. And this is the paradigm, I think, with which as CWL is built. There is a lot of like a very explicit setting of what should be executed and what is the order of steps. And I think it is at first it is a little easier for people to start. At least it is evident basically what is the order of processes. But at the same time, this also comes at the cost of that the syntax of the language is basically more complex itself. At least in my view, there are more elements to it, and more different things you have to keep in mind, again, in order for all of the necessary details to be included in there. Snakemake is, at least at the time when it was developed initially, used only the implicit paradigm, which implies that basically the sequence of steps is determined by just the matching input and output file. So I think I can show what it looks like. So take a certain Snakemake workflow. So, for example, I have some over here. Yeah, this is not the best example of a Snakemake workflow, I shall say, but that's some of the early things which we've been doing. Yeah, then we have, let's say, like a couple of processes, like sorting of the BAM files after read alignment, and then marking the duplicate reads. And basically, we have the output file, which is set here, like this. Yeah, the output of the sorting is the BAM file, which has this like sorted.bam suffix. And this is the actual file, which is stated here as input. And Snakemake, you don't have to set manually that this particular input comes from this particular process. So as you can see here, I do not provide any reference to sorting of BAM files to the previous step. basically there is only the reference to the input file, and then the Snakemake would automatically go and take the from other processes, other rules, the one which has this file as output, and will decide that the sorting of BAM files has to be executed before marking the duplicates. And this is the idea of implicit description of workflow, in which you don't have to set, again, clearly and explicitly that this step comes after this step. And there, in terms of, again, defining your processes, this is a little bit easier, but this in turn comes at the cost of a pipeline being less readable, less understandable for people who are not familiar with what's going on. There is a convention of writing these rules one after the other in the order they are executed, but still, again, there is no direct reference to the exact process. Yeah, returning back to the question of why we've chosen Snakemake at the time, we've chosen it for the reasons of basically being simpler to write and use with all sorts of different tools, because CWL, in addition to it having a more complex syntax, it also has to be, so for each software tool which is there in bioinformatics, it usually has to be defined in terms of a specific file, like configuration file for CWL, which tells it how to use the tool. So basically, you cannot just specify a shell command as we do here in the Snakemake workflow. We basically just show the shell command in here to tell what should be executed. For CWL, you have to define, again, you have to wrap the tool into the CWL code in order to be able to use it. And basically, this is the main reason, was the main reason. Now Snakemake, I think, includes functions for setting the explicit connections between rules, so you can write down that a particular input file is the output of another rule. So that this was not possible at the time when we started using it. It's, I think, possible now. And it also now includes the wrapper functionality, which is pretty similar to CWL. So some tools in bioinformatics are already inserted into basically the standard repository of Snakemake, and you can basically use these wrappers to run, for example, like GADK. You will not have to write down the exact command which has to be executed in the shell. It will basically be defined within the Snakemake itself. So that's also a nice feature which they've added some time ago. So this indicates that the languages themselves are developing. Yeah, but still, I think that Snakemake is still simpler for people to start writing with that. It is interesting that some people say differently. Some people say that actually Snakemake is the toughest one to study, which I don't think that that's the case. I think that, again, given the relative simplicity of the syntax, it is not very complicated to start writing the workflows with Snakemake. Another nice thing about Snakemake is that it can make this visualization. So, for example, like this, you may have all of the tasks which are being performed visualized as a graph. So this is a so-called DAG, or directed acyclic graph of jobs which are executed by Snakemake. And you have basically here the order of steps. The dashed lines over the circles mean that the output files have already been generated. So this is the step which actually will not be run if you execute the pipeline, because as with most of the pipelining languages, they are capable of determining which files you already have, not to run all of the steps from the beginning. And Snakemake can also do that. And again, this nice visualization. I think that I didn't see the same functionality for Nextflow, for example. Maybe I was not very attentive, not studying the documentation in much detail. But I think that Nextflow doesn't have this option, and this is really nice to see the actual order of tasks for all sorts of purposes. Yeah, so that's basically I think the Snakemake for us was the choice due to relatively higher simplicity and compared to the options which were available at the time. Now again, with the availability of Nextflow, I think that the choice might be a little bit harder, but still, Nextflow, in my perception, is still a little bit more complex in terms of getting started with it and working with it compared to Snakemake. Yeah, so that's my take on the matter, and I think if there are any other people like in the audience who have responded, for example, to our quiz that you use Nextflow, yeah, I think that there were several people. We now have 14 responses. Yeah, and we have three people who have used Snakemake. Not Snakemake, Nextflow. I'm sorry. So if any of the in the audience who've used Nextflow could comment on basically the reason of the choice of why you use Nextflow, that maybe you have some ideas regarding the advantages and why do you prefer this to other options which are available.
I believe the one possible solution is that if you have cluster with installed Nextflow and don't have other solutions on this cluster.
Yeah, that's a nice thing. I wonder whether there are any other reasons. Yeah, I believe there should be, because it's quite popular again regarding what I mentioned that Alex Predil said that like Sangar Institute uses Nextflow mostly for production purposes. So I think there should be some benefits to choosing it over the others.
Yeah. Unfortunately, I have no example with Nextflow language and haven't touched it before, so can say if in this case. Probably we need somebody who familiar with Nextflow engine.
Yeah. That's why I'm asking. So if there is anybody in the audience, and there are people in the audience who are familiar. So yeah, if someone can comment, if not, then that's okay. Yeah, we also have, I think, in the quiz here, one people who have responded. That's the next response which appeared in here. That some people used graphics or web interfaces for pipeline description. Yeah, so I think that might be an interesting case as to what was this option. So again, also if someone from the audience could comment on what basically was this graphics interface or web-based platform for pipelining, it also be nice. You may very well share your opinion in the chat if you have no opportunity to talk for this or that reason. Yeah, but that would be really interesting to see, because with this graphic things and the web platforms, that's usually an idea which is more or less evident, I would say, because the pipelines are naturally like this basically workflow diagrams when you have to connect process one to another. So the idea of constructing something using basically just this idea of having blocks joined to one by one is a nice idea, and I think there were some platforms that provided this functionality. I'm not sure as to why they are not that popular. I think Alexander, do you remember what was the name of the platform? I think you worked previously. Okay, the 9, yeah. 9. And do you have any comments as to what was what was your impression? Because you are the one.
Actually, it's pretty useful when you finally understand how does it work, but it's not too simple.
Okay. Yeah. So the obvious benefit, I think, from the graphic things is that they have a lower, let's say, like entry barrier. So if you are not that familiar with basically coding and writing some software, it will be much easier for you to start with the platforms like this. Yeah, but still they have to, as Alexander just said, you have to have some sort of tutorial as to how to use them. But yeah, KNIME, I think it's like KNIME. This was basically the one of the platforms which provide this functionality. Yeah, and it's also like nice to see that no one has still no one has mentioned like using either CWL or WDL. Yeah, and also no one used Apache Airflow. I think I have to comment a little bit on that. So as to what was the option and why I've added this. So basically, Apache Airflow is a very specialized solution. You don't have to confuse it and Nextflow, because Nextflow is a pipelining language, and Apache Airflow is a workflow system which is designed for so-called Apache clusters, or like Spark clusters. So basically, the Spark cluster is a very specific thing where you have not one server but multiple servers which are united into a single cluster with the so-called logic of distributed computing and distributed file system. And when now things are basically scattered across multiple units, and then each unit processes its chunk of data separately. That's like a MapReduce framework, if I'm not mistaken the words. And again, the Airflow is a very generic thing. It's not specifically designed for bioinformatics, but again, I know people who have used Airflow in their practice to develop pipelines for production on Spark clusters. So it was nice to see whether some people in the audience have this experience, but it seems that no one had. Oh, that's okay. Okay, good. So if there's no one who would like to tell us anything about the motivation of preferring Nextflow, then I think we may move on. I have two more short questions in this quiz, which I've asked you. So first was about whether the pipelining languages are considered to be complicated, and basically. Okay, it keeps refreshing. I wonder why. Yeah, and most of the people actually say that. I think not more, but still the majority of the people who have answered this question say that they are not too complicated. But there are 3 people who say that they are. I think I may add to this question that. This is, I think, for many of the options which are available, like CWA, for example. That's really it may be a really tough task to get to get started, to have your like first experience with that. And but for the other options, I think that they're like more or less friendly, especially for people who are familiar with basic programming. So, for example, again, Snakemake uses many of the logic of the Python, and you can basically just write Python code inside the Snakemake workflows, which is also pretty useful in many cases. So yeah, that's probably not the case for Snakemake in my view, but for many of the options, it definitely is. Yeah, and the last question which we've had in this quiz, which I think is also very important in terms of pipelining solutions, is that most of these pipelining languages have repositories for community developed pipelines. So for example, with Nextflow, you have the so-called NF core, which is a very large repository of pipelines which are developed and maintained by people in the community. So I may even show you, well, basically what it looks like. So you basically have, again, this is the NF core. So if you go, let's move on, move backwards. Yeah. So this is basically the NF core website, And in here, you may basically just browse all of the pipelines which are implemented in here. And the first thing which is shown here is the Sarek pipeline, which is quite nice. That's the thing which is used to run GATK variant calling. Again, indicative of that people don't usually like to use the GATK's own pipelining, like workflow languages, which is I can confirm that that's a little bit weird. So I can understand people switching to other options. Yeah, but basically, yeah, here you have a long list of all sorts of pipelines which have been developed with Nextflow. So for example, with Sarek, you may go over here, like look into what it does, basically what are the input and output files, what is the sequence of steps which is being performed, what is the logic in here. And you may also go to basically just the repository in GitHub. I hope that all the people who have been on our first meeting, even if you have never worked with GitHub yourself, you at least saw it last time. Yeah, but basically here you have this description of a pipeline in terms of like of a Git repository, all sorts of information about this, and you may just like take it and run it. Very simply and very easily, and I know again people who have done so, and their motivation was like this. So I know that this is a community maintained thing. I don't care what happens inside. I know that it works and many people use it, and that's why I will use it as well, because I trust people from the community more than I trust myself. So basically, this was the kind of logic which was which people whom I talked to who used this exact Sarek thing, they were telling this. And but I myself have some doubts regarding the trustworthiness of the pipelines for public repositories. So I would like to hear your opinions. For example, again, Gena, do you have any opinions regarding whether pipelines from basically like this public repositories, are they trustworthy? Do you trust them personally or not? And if so, what's your take on that?
I have the 30-second answer to this question. Probably I will trust in case that I will probably try to run it and follow. But in fact, I have not so much experience with such pipelines. In fact, I haven't met any pipeline which can be run as is. I still had to look at it, walk through the code, just to make it work. So, in general, the answer is yes, that it should do the things what it was designed for, but I still have to make a lot of tuning and something like this.
Okay, yeah, so that's reasonable. I think, yeah, that because especially if you're using it for something important, and again, this Sarek thing is used for variant calling in medical genetics. Again, I spoke to people who have been running it for medical genetics purposes. I think medical genetics is a thing which includes quite a lot of responsibility. Because again, if this is for diagnostics, you have to be sure that you're doing everything right. And yes, reviewing what you are using is a very important step. Yeah, but generally, yeah, if that turns out that it behaves as expected, if there is nothing strange in there, you may very well use it. Yeah. Roma, do you have anything to first? Did you have any experience of working with these like publicly available pipelines? Have you used any of that or not?
Yes, I tried to use. So for Snakemake, there is a repository with curated list of workflows. So it doesn't mean that these workflows are like best practices. So everyone could submit his workflow there and created. That means here that authors of Snakemake, they validate that workflow is well designed, like use all necessary concepts of Snakemake and the right directory structure and so on. So I tried to use some of these workflows, but for me, so it didn't work well. So I need to slightly modify it and change for my use case. And you mentioned that there are wrappers in Snakemake, but recent Snakemake version, so wrappers are basically a very convenient way not to think of command line arguments and how to install tool. So they provide you a nice wrapper for that. And the recent versions of Snakemake, like it was introduced maybe a year or 2 years ago, they allow you to reuse some publicly available workflows like this workflows. So you can basically you can extend workflow and do not copy it, but just modify some of steps or some of rules. So all these workflows are open source. So my answer was that I don't trust any existing workflow before I have to verify it, check what it does. Is it okay or not from my point of view and according to some published literature, and only after then I reuse it or maybe use as example for my workflows.
Yeah, that's thank you. That's actually the kind of approach which I completely endorse, because that's I personally have I think never used the workflows which are published as is, but yes, I've used them for like as a reference in some cases, basically taking some steps from things which were published previously. Yeah, I'm not, in fact, a big fan of just taking a thing and running it. I usually tend to criticize people who do it this way, because again, with pipelines, it is slightly different from the tools in the sense that if you have a software tool, which is for example, an aligner or something like this, usually it is quite tough to read through it and understand what it does and whether or not it is reasonable. But in terms of the pipelines, it usually doesn't take too much time to look through it and to understand whether or not that's reasonable. Yeah, and that saves you time probably to take it as is after you review it, and that might be okay. But I completely agree with both people who have spoken so far that doing your own review of what's in there is very important to assure yourself that nothing will go wrong with it. Yeah, okay. So if anyone else has again from the audience, I saw that German, I think, was here. I'm not sure whether he mentioned that he is expected to drop. Yeah, so. If anyone else again here has anything to add regarding either the choice of language or the trust in published pipelines, or we've had an issue of last time we didn't have anyone to tell anything regarding the benefits of Zenodo, etc. And now I think we have the same issue with Nextflow. No one to advocate for it, unfortunately. Yeah, again, if anyone has anything to add to the discussion so far, then please go ahead. Or if you have any question related to that. Okay, so it seems that we don't have any comments and any opinions so far. Then we, I think, conclude that, and most of the people actually agreed that these things are more or less trustworthy. I think we've agreed that you can use very well the published repositories, workflows, but you have to review it if you want to assure that your analysis is of high quality. I think that most of the people who have spoken today have basically spoken in favor of using Snakemake rather than Nextflow, which is a little bit of a pity, in my opinion, because it's not good that they didn't have anyone to defend the Nextflow. But again, it gains popularity quite significantly recently, so that's probably a reasonable place to consider it if you are choosing a language for your own purposes. Yeah, and we've I think discussed quite nicely the benefits of using the languages and the advantages over typical like shell scripting, etc. And reproducibility of this and reusability is a very important thing. Okay, so having said that, then I'd like to thank again everyone for participating in today's discussion. I have to tell you that next time we're expecting more people to join us with the expert panel. I hope that this will make our discussion more productive and more interesting, probably. Yeah, and next time we'll be diving into the issue of dependencies and organizing the data analysis. So we'll talk about virtual environments and Conda, both virtual environments themselves and Conda as a package manager, because there's a fair lot of things to be discussed there. And again, we've had several notable people invited for the next time, so I hope the discussion will be nice. Yeah, and the time after the next one, we'll talk about containerization. Yeah, so next meeting is on 22nd of June. The containerization will be, I think it's like 6th of July, if I'm not mistaken, the numbers. Yeah, so then thank you very much for your participation in the discussion today. And
Yura, one comment. So seems you forgot one of the questions that you wanted to discuss, that about should we update tools in our
thank you so much.
workflow or not?
Yeah, thank you so much. Yeah. So before we end today's discussion, yeah, actually, when we were preparing for the seminar, we've discussed also that we wanted to talk a little bit about the updates. Yeah, because I've raised this question last time when we were talking about data sharing, about the updating of repository, whether or not it's okay in terms of you basically changing the data which are associated, for example, with the paper. And yeah, the pipelines, it's also quite an important topic, because they have pipelines. If you want to achieve reproducibility, on one hand, you probably want your versions to stay the same, version of software you use to stay the same over time, in order to compare what you've done like some time ago and what you're doing today. But at the same time, again, yeah, updates of software, as you know, may bring very useful changes, fix some problems, and that's makes this question a little bit of debatable. Yeah, and yeah, so Roma, I think you remember that because you had something to say about it. Yes.
Yes. So sometimes we try to update tools. For example, MultiQC. It's good to update because new version of MultiQC adds some support of additional tools and additional cool reports. But recently we faced with one drawback. So one of our tools for methylation analysis, which is MetPipe. So we decided to update it because according to their GitHub page, MetPipe was was deprecated, and also asked to use their new version, which is DNAME tools. But the funny story was that this change was should be poor technical, so they refactor the repository and didn't change the algorithm, but unfortunately they introduced a very weird bug. So in their differential analysis, all most strongest p-values were just erased, and only weak p-values and average p-values were left. And we noticed, we noticed this issue because we compared like our results with some previous results with some common sense. And so we identified the problem. So please, so it's maybe sometimes it's good to update your tools, but be very careful with it. Maybe compare results and with different heuristics with previous version, with some common sense, because even in some long existing and cool tools, errors could be introduced easily.
Yeah, thank you. Actually what you've just mentioned made me remember a thing. I am struggling to find the exact issue. So basically, there is a tool which is called BWA MEM2, which is a re-implementation of the BWA MEM aligner. Again, the BWA MEM aligner is of the publicly available ones, just basically the best performing thing you can find, especially in combinations with some of the variant colors, and it's also the most used one. This MEM3 implementation, it actually uses some CPU level and advanced technologies to basically speed up the alignment, and it does it like I think 2 to 3 times. So this thing works 2 to 3 times faster compared to the original BWA MEM. And we've included this into some of our pipelines when it was the version I think 2.0, and then at some point in time there was an issue reported. Again, I think I cannot find it right here, but still there was an issue that there was a major discrepancy after a certain update. There appeared a major discrepancy between the results of BWA MEM2 and the original implementation of BWA MEM. Again, it is used for the most important use case of BWA MEM algorithm is for medical genetics. Naturally, that's a large problem. Fortunately, we didn't update the pipeline that regularly, so we didn't have this issue. But yeah, I know people who have suffered from that because at a certain version, everything was okay. The results were perfectly matching, and then they stopped matching, and there was an issue of a high discrepancy. Yeah, I think that there is fair lot of issues. So, for example, there is some very recent issue that the output like basically is different between MEM and MEM2. This is again the recent issue. I think no one has answered that so far. But this is not the one which was basically at the time, which what I was talking about. But still, yeah, this is the issue with updating. So probably if you are working, my take on that if you are working with some, especially if you have a production pipeline, which you are not using basically much for ordinary research purposes, but if you are using it for, let's say again, as in diagnostics or in other important tasks, where you provide some output to clinicians or to other people who make decisions based on your output. Then when updating the tools, you have to look into the versions very carefully and consider, so not just basically taking the newest version when it becomes available, but maybe waiting a little bit to see whether there will be any issues reported, maybe comparing the results in terms of whether they look the same or not. Because unfortunately, yeah, updates sometimes disrupt something. Again, thank you, Roma, for the very nice example. Again, this BWMM thing is yet another example of the same issue. So yeah, if your data analysis leads to some important decision making for some people, then we advise you really to take care of that and update only to some stable versions, not do it, but basically just anytime you see a newer version is there. Yeah, and also for the medical purposes, there are also guidelines for this case. So for example, for medical genetics, you have guidelines which advise to test the pipeline with a standard sample each time you update anything. So this is also pretty important. I think that if there are updates which look nice, which claim to have introduced some benefits, some improvements, it probably is a good idea to rerun your pipeline with some standard sample each time you do these updates, to basically just make sure that nothing is broken, that you don't have any weird results in the end. Yeah, that's thank you, Roman, for the reminder to discuss this issue. Yeah, thank you for the comment. Okay, good. Then now I think it is time for us to finish for today. Again, thanks everyone who's participated. Next time, a heated debate on environments and conduct. I hope that will be a heated one. Again, saying that we hope more people to join us for the expert panel. Yeah, again, thanks to everybody who has joined us, and goodbye.